All right, class. So this is chapter 9 on sensory systems, and this is the last chapter of this module or unit. So after this, you can take the lecture exam. We'll be going through your different sensory organs. First of all, we'll talk about sensory receptors. These are structures that are specialized to detect and respond to changes in the external or internal environment. If a stimulus is strong enough, then action potentials will be conducted to the brain from these sensory receptors. They allow for sensation, which is just awareness of a stimulus, and how a sensation is experienced, such as sight or sound, depends on which part of the brain receives the impulse. Perception is the conscious awareness of sensations, and understanding the stimulus occurs when the cerebral cortex integrates sensory input. So here's an example of how this works. Some sort of stimulus, whether it's touching something or seeing light, will be detected by a sensory receptor. This is a Pacinian corpuscle receptor in your skin, and this is just detecting light in your eye. That nerve impulse is sent to the brain by sensory nerves, brain or spinal cord, and then the brain and spinal cord will interpret that information and then send out some sort of response. Sensory adaptation is when sensory receptors stop responding when they're continuously stimulated, leading to a decrease in the awareness of a stimulus. This can vary among receptors. Pressure and touch receptors adapt quickly, but receptors in muscles and joints that report on body position never adapt, which is good because you will always want them telling you where your body position is located. Here are specialized receptors, and we'll go through these. General senses include touch, pressure, vibration, temperature, body and limb position, and pain. Receptors are located in skin, muscles, bones, joints, and internal organs. We're not usually aware of the general senses, but they are still important. They provide information about body position and help keep the internal body conditions within optimal limits. The special senses then are the 5 that you probably have learned about since you were in first grade: vision, hearing, equilibrium, which is also involved with your ears, smell, and taste. And we rely on these special senses to perceive the world. Receptors for the special senses are located in the head, often within specific structures like the eyeball or the ear. Receptors rely on either free nerve endings or encapsulated nerve endings, which are just the tips of sensory neurons or dendrites, are the free nerve endings. And encapsulated nerve endings are those in which a connective tissue capsule encloses and protects the tips of dendrites of sensory neurons. So here's an example of a free nerve ending in your skin in the detecting pain, heat, and cold, wrapped around hair. Here's a Merkel disc, which picks up light touch sensations. Here's a Meissner's corpuscle that helps pick up light touch. Here's a Pacinian corpuscle, which is located deeper in the dermis of the skin that picks up stronger pressure. And here's a Ruffini corpuscle, which picks up continuous pressure. So these are just different pressures in the skin. Touch, pressure, and vibration rely on mechanoreceptors that respond to stimuli that stretch, compress, or twist their membrane. So mechanoreceptors detect touch, and there's different ones found in the skin. We have Merkel discs and Meissner's corpuscles. Merkel discs are free nerve endings that detect light touch, and encapsulated nerve endings are the Meissner's corpuscles that tell us where we have been touched. Different mechanoreceptors occur in the skin that detect pressure. Pacinian corpuscles are layers of tissue surrounding a nerve ending that sense the first pressure or vibration. And Ruffini corpuscles are encapsulated nerve endings that respond to continuous pressure. So a lot of nerve endings are found in your skin just so that your body's able to sense any sort of external environment. It's kind of your body's first line of defense. Thermoreceptors respond to temperature changes. They can adapt rapidly. One type will respond to warmth and another type will respond to cold. Body and limb position are continuously monitored by the brain as muscle spindles, which are specialized muscle fibers wrapped in sensory nerve endings. They monitor the length of the skeletal muscles, Tendon organs are highly branched nerve fibers and tendons that measure muscle tension, and they also, information from the inner ear helps with body position. So here's the muscle spindle sensory neurons responding to the tension in a muscle, and a tendon receptor responding to the tension in a tendon. Free nerve endings found in all tissues of the body can be damaged that release chemicals that alert free nerve endings. Aspirin and ibuprofen interfere with the production of these released chemicals, so they help to decrease that pain that is perceived. Referred pain is pain felt somewhere besides the site of the injury, and it's common with damage to internal organs. For example, here's a look at referred pain. If your appendix bursts, it could be referred to not just the location of your appendix, but anywhere on the right side of your body. If you have pain in your colon, it could be referred to just the central of your stomach. The big one is your heart. If you have a heart attack or pain in your heart, the pain could be referred all up and down the left side of your arm. So let's talk about vision and the eye. The outermost layer of the eye is called your sclera and cornea. The middle layer is your choroid, the ciliary body, and the iris. And the innermost layer is your retina. The outer layer is tough and fibrous, so the sclera is the white part of your eye. It protects and shapes the eye, and it serves as an attachment for all the eye muscles that allow movement of the eyeball itself. The cornea is transparent. It's the part in front of your eye that you would place your contact lens, and it allows light to enter. The middle layer is vascular. It's your choroid layer that contains blood vessels that supply your eye with nutrients and oxygen. The ciliary body is a muscle that encircles the lens and holds the lens in place to change its shape. And the iris is the muscular part of the choroid in front of the ciliary body. It regulates the size of your pupils, and the iris is the color of your eye that we all have different colors for. The pupil is just a hole in the center of the eyeball. So it's the black opening in the center of the iris, and it allows for light to enter into the eye and reach the back of the eye, the retina. It'll dilate or become larger in dim light, and it'll constrict in bright light or become smaller. The retina is the innermost layer of the eye. It'll contain special photoreceptors that respond to light. And these two types of photoreceptors are rods and cones. And the fovea is an area in the retina with the greatest concentration of cones. So that just means that your eye will work to focus whatever it's reading so that the images get transmitted to the back of the eye and lie directly on the fovea, because that's the area where the cones will help see it. So we say that objects are focused here for sharp vision. Your eye carries visual, or your vision involves the carrying of visual information from the eye to the brain for interpretation. Each one of our eyes has what we call a blind spot, where your optic nerve leaves the back of the eyeball at the retina, and the retina lacks any photoreceptors here. So if an image strikes that blind spot, it cannot be seen. And if we were in class, we would do an activity to demonstrate, and you'd be able to see the blind spot in each of your eyes. And then within the eye, you have a posterior chamber and an anterior chamber. The posterior chamber is located between the lens and the retina. It contains vitreous or jelly-like fluid. And the anterior chamber is located between the cornea and the lens, which contains aqueous humor. It's more watery and it's continuously replaced. So I've talked a lot about the eye, and now we can just take a look at it. So here's the iris controlling the shape of the pupil, the hole right through it. The sclera is the white part of the eye. The cornea is this transparent piece that will cover both the pupil and the iris. You can see your aqueous humor filling your anterior chamber, and the posterior chamber filled with vitreous humor behind or in front of the lens, respectively. Here's the lens, and the shape of the lens, which allows you to focus images, is controlled by the ciliary body, this muscle right here. You can see the three layers of the eye, the sclera, the choroid, the retina, and then you can see the area where the optic nerve attaches to the eyeball, which will bring all of that sensory information to the brain. And the blind spot is the optic disc, the area where the optic nerve attaches, where there would be no photoreceptors. And the fovea centralis is that area shown right here, and that's the area of sharpest vision. So that's a little about the eye. Glaucoma is the second leading cause of blindness, and this results when pressure within the eye reaches dangerous levels. It results when drainage of the aqueous humor is blocked. So pressure builds up, blood vessels supplying the optic nerve and retina collapse and die. And it can be really progressive and actually extremely painless. So that's glaucoma, a buildup of pressure within the eye. Sharp vision requires light to be focused on the retina, and light is bent or refracted at 4 points when it enters the eye. It's refracted at the cornea, through the aqueous humor, the lens, and through the vitreous humor. The lens is elastic and can change shape. The ciliary muscle controls that shape. We call accommodation the changing of the shape of the lens to change the bending of that light. And a cataract is a lens that has become cloudy, usually due to aging. So here's a look at how the shape of the lens allows you to see things close up or far away. Everything that we do see actually gets rotated, and we look at it becomes upside down in the back of the retina, and then our brain is what puts it right side up. For a close object, the lens becomes more round, and for looking at things far away, the lens becomes more flat to allow things to focus on the back of the retina. And here's a look at someone with a cataract. Focusing problems, you can be farsighted, nearsighted, or have astigmatism. Nearsighted means you can only see things far away. Farsighted means you can only see things near. Causes include discrepancy in the thickness or curvature of the eyeball. Normal vision can be restored with corrective lenses or LASIK. So farsighted, you can only see things far away, so you can see distant objects more clearly. Nearsighted, you can see nearby objects more clearly. So most of us who wear corrective lenses are nearsighted. We can see nearby things more clearly. Astigmatism has to do with the shape of the lens. So it usually means your lens is too steep, and it'll distort the visual image and make things a little blurrier than it should be. So here's a look at farsightedness and how farsightedness can be corrected. So you usually have what we call a convex lens. Nearsightedness, myopia, also known as, can be corrected with a concave lens, and that's the shape of our glasses or contact lenses. And astigmatism can also be corrected with a contact lens. So light is focused on the retina, and it causes changes in the photopigment molecules within these photoreceptors. The photoreceptors can release less inhibitory neurotransmitter, and the activity of cells processing visual information increases. So electrical information from those rods and cones is sent to bipolar cells, then to ganglia cells, and then to the brain. So here's a look at how things get focused in the back of the eye. They are picked up by your ganglia cells, then bipolar cells, then your photoreceptor cells, and then they are picked up by the brain. Two types of photoreceptors, rods and cones, respond to light with a neural message. Rods are important for seeing things in dim light. They are more numerous than cones, and they are also responsible for black and white vision. They contain the pigment rhodopsin, so that's why we call them rods. Cones are responsible for color vision. There's blue, red, and green types of cones. They produce sharp images, and a reduced number or a lack of one type of cone will result in color blindness for that appropriate type of cone. So here's a look at what rods and cones look like under a scanning microscope, and you can see that cones are more conical in shape, and rods are a little more parallel in shape. This is a look to test for color blindness. You might see pictures like this in a book testing you for color blindness. So hopefully you can see the letter 12 in this picture. If you can't, you might be colorblind to the color red because the 12 is in red dots. Here's a great look a video to review the anatomy of the eye. In order to hear, the ear collects and amplifies sound waves that are produced by vibrations. The loudness of a sound is determined by the amplitude of the sound wave. And the pitch of a sound is determined by the frequency of sound waves. So here's a look at low versus high amplitude and low frequency versus high frequency, giving rise to differences in loudness and pitch. There's three main parts to the ear: the outer, inner, and middle ear. The outer ear functions as the receiver, and it includes the pinna, which is the outer ear that you can feel, and the external auditory canal, which is just the area of the ear canal that will lead to the tympanic membrane or the eardrum. The tympanic membrane then separates the outer ear from the middle ear. It vibrates at the same frequency as the sound waves and transfers these vibrations to the middle ear. You must have nearly equal pressure on both sides to vibrate properly. And the auditory or eustachian tube connects the middle ear cavity with the throat to help alleviate pressure differences at high altitudes or when you're in airplanes. The middle ear then functions as an amplifier of that sound. It's an air-filled cavity in the temporal bone of the skull, and it contains these three auditory bones, the malleus, incus, and stapes. The middle ear bones can convey sound waves from the eardrum to the oval window, which is just a sheet of tissue at the entrance to the inner ear. And the force of the eardrum's vibrations is amplified 22 times in the middle ear. And this helps to transfer those vibrations to fluid of the inner ear. The inner ear then functions as a transmitter. It generates neural messages in response to pressure waves caused by these sound waves, and then it sends these messages to the brain. It contains two sensory organs, the cochlea and the vestibular apparatus. The cochlea helps with hearing, and the vestibular apparatus helps with body position and movement. So this is a great look at the anatomy of the ear. You can see the outer ear, the middle ear, and the inner ear. So I'd encourage you guys to learn this as well. The cochlea looks like a snail. It has two openings: the oval window, which is what the stapes fits into and transmits those sound waves into fluid waves, and the round window, which relieves pressure. There's three longitudinal compartments filled with fluid within the cochlea if we were to unravel it. The central compartment consists of the basilar membrane, and the spiral organ is the organ most directly responsible for hearing, and it consists of teeny tiny hair cells that will bend into an overhanging tectorial membrane. So this is a look at the cochlea, and if we take a cross-section of the cochlea, we can see that it's made of three different chambers. And if we zoom in on this area, this is the organ of Corti, the spiral organ. This contains those teeny tiny hair cells that when pressure waves from the sound will cause them to bend, and the bending into this tectorial membrane will be picked up by the nervous system, which will send those bending simulations to the brain. Here's the sequence of events in the inner ear that I'll let you guys read through as the process of hearing occurs. Loudness, the louder the sound means more hair cells are stimulated as the hair cells bend and increase the number of impulses, and the brain will interpret the increased number of impulses as louder sound. Pitch is sounds of different pitch activate hair cells at different places, and the brain will interpret input from hair cells in different areas as sounds of different pitch. Video on hearing that you can watch. There's types of hearing loss, whether it's conductive or sensorineural. Conductive involves an obstruction along the route that sound follows to the ear, and sensorineural is caused by damage to either the hair cells or the nerve supply to the inner ear. An external ear infection is infection in the external auditory canal. It can be caused by water trapped in there due to swimmer's ear. It just makes a great environment for bacteria to grow. A middle ear infection is usually the result when infections of the nose and throat move through the auditory tubes to infect the ear that way. And it's more common in children because their auditory tubes are horizontal, allowing easier access to bacteria. The vestibular apparatus then is a maze of fluid-filled chambers and canals within the inner ear. This helps with balance, and the semicircular canals specifically help with balance when we are moving, and the vestibule helps with balance when we are not moving. The semicircular canals are 3 canals in each ear responsible for dynamic equilibrium, and movement of the head causes fluid within these canals to move, which pushes and stimulates these hair cells. The vestibule consists of the utricle and the saccule, which sense forward tilting of the head or vertical movement of the head, respectively. They're also responsible for static equilibrium, so when these hair cells move, those messages are sent to the brain. So this is a look at semicircular canals and the utricle and saccule within them and how they respond to head facing forward, head facing left, head upright, or head tilted. Then we'll get to smell and taste. Your olfactory receptors help with smell. They are in the nasal cavity. The odor molecules will dissolve and bind to these receptors, stimulating them, which send on that information to the brain. So here's a look at smelling picked up by olfactory sensors and sent through the olfactory nerve to the brain. Taste buds are located on the tongue and the inner surface of the mouth. They're replaced every 10 days, and here are the 5 basic tastes that we can taste. They have taste hairs that project into a pore, and these taste hairs have receptors for food. When food molecules are dissolved in saliva, they enter the pore and stimulate these taste hairs. Although they're not neurons, these taste cells can still generate electrical signals that are sent to sensory neurons wrapped around them. And here's a look at taste buds found in the papilla, these little tiny bumps of the tongue. So from this chapter, you should be able to, again, use this checklist to make sure you covered everything that you should know. Thanks for listening. This is the end of chapter 9 and also the end of our second module and second unit.